Okay, hi guys, welcome back. So when I say welcome back, I think it's a bit more to the YouTube students because I'm actually uploading this into YouTube and sharing with my existing I think about 700-800 of my Form 5 students. So if you have been watching my YouTube for a while, and you find that, hey, Mr. Khor, what happened to you? How come like the whole, don't know how many months you've been quite inactive? Is because I had too many things that are ongoing. My existing students know. In fact, 2 days later, the day after tomorrow, I'll be giving a bio talk under Datuk Seri Anwar Ibrahim, a bio talk, not political talk, a bio talk to his students around Tambun. 15 schools are invited. 15 schools are invited. We expect to have a crowd of 700 SPM, calon SPM 2022. Okay. Okay. Anyway, guys, if you saw the title, this video is about recapping paper 3 a bit. I've been getting a lot of messages about paper 3 of late. I mean, common sense. So about next week is already paper 3 starting Monday. Monday, I think 30th you have physics, then 31st you have bio. You skip one day, then after that Thursday is chem. Okay, so guys, let's get right into it. Let's get right into it. Yeah, okay. Okay, so guys, what we are looking at here is Terengganu paper 3. Terengganu paper 3, this is where I think it's not a huge secret that there were a few lists. There were a few lists, meaning the apa senarai radas dan bahan, a few lists that was out. So in short, everyone is expecting fermentation of yeast to come out. Okay, it's not a secret, guys. It's not a secret. Oh, sir, I do not know whether you will see or not. But I think most of the students, even teachers, are expecting yeast to come out. I told a group of students in online seminar that day, around this time, there's a lot of penilit. Have you heard of penilit nasib? In English, we call it fortune teller. 那种看命的，看命 fortune teller 啦 fortune teller. Okay, around this time, the whole country got suddenly a lot more fortune teller, and these are existing teachers. Yeah, this is something that happens a lot around this time nearing SPM, guys. It's not yeah, it's a norm, I guess, in Malaysia. Yeah, you know, certain things only happen in Malaysia. So around this time, there's a lot of teachers who will try to predict what can come out, but the intention is the same. In order to help you. The intention is to help you, untuk menolong Anda dapat grade yang bagus dalam kertas 3. Yeah, there's no bad intention. When a teacher tells you something, definitely I bet he or she wants to help you. Okay, but what I am more worried about is you only read one experiment. Itu kerisauan saya. Yeah, my worry is that you only focus on one, the rest you don't care. So I think we need to always buffer a little bit. Okay, so guys, don't only read fermentation of yeast. Try to go more than that. Okay, try to go more than that. You can take note. Oh, this teacher say this will come out, that teacher say that will come out. It's okay, guys, because it's just experiment. It's not a lot to study, frankly. It's not a lot to study like what we have in our bioscope. Okay, So what I'm trying to tell you is your teacher have good intention, but sometimes fortune teller can tell you a lot of things, but sometimes it may not all be true. Okay. Even I told my students, even myself, if I tell you that yeast fermentation can come out, these are roughly the steps. I told them 100% it won't be the same as what I'm telling you. Because why? Experiment is very specific. So even in my WhatsApp group, if you are my existing student, I told you, take with a pinch of salt. I hope nobody took it literally lah. Oh, cikgu cakap kena makan sikit garam. I really hope nobody did it lah. Yeah, I put that, take with a pinch of salt, meaning have a look at it, read up, get to know, but still cover some other experiments. Yeah. I think I better tell them the next class. Yeah, ask them who took some salt that day after I posted in WhatsApp. Okay, so mari kita mula. Guys, so these are the lists that they gave, very similar to probably what you have obtained from your WhatsApp, from your teacher via WhatsApp or maybe even Telegram. So now, to me, two things that are important. I think the main thing, the main thing that's very important for today is how to tackle questions, how to tackle and answer those questions. Okay. I think 2 days ago, a student, not even my student, Mr. Khor, can I ask you something? I said, yeah, of course. In IG, in my Mr. Khor Biology IG, my Instagram, someone asked, sir, can I ask you a question? But I'm not your student. I said, can, by all means, come, you can ask. Sir, can I know how to answer observation, hypothesis? Wow. I told her I will upload a video, so you check it out then. Okay, so this video is basically for whoever that's watching, can be my student, can be YouTube, can be you share out. I'm okay, guys. I got no issues with that. Okay, let's start. So for paper 3 bio, kertas 3 bio, the questions that are generally asked, these are the few. If you have followed me for quite a while, I mean my normal class students, you know that I emphasize on this 11. There are 11 questions that they can ask. The very first one, because you are doing it hands-on, ini eksperimen practical, ini amali. Maksudnya kamu kena mengendali, boleh ubah. You have to handle the variables. So once you get the value, you have to write it down. Apakah nilai? What's the value? How many pH? How many cm cube? Yeah. The numbers are involved. Then from here, they can ask you observation, pemerhatian. Then normally, if got observation, they will link to inference. Okay, hypothesis is nothing new. They can ask you table. They can ask you to draw a table. Sorry, I think I better put variables first. They can ask you to draw a table. From the table, construct a graph, then get to know the relationship. Okay, then prediction. Prediction number 10 would be operational definition. Guys, definisi secara beroperasi. And number 11 here, I call it classifying items. So what do you mean by classifying items? They give you example, a list of items, you have to classify. Oh, biotic factor, abiotic factor. Or something very simple would be apparatus and material. I think they don't ask that anymore. Or they can ask you metal ion, non-metal ion. Okay. They asked in bio before once, metal ion, non-metal ion. Okay. Okay, so guys, these are the things that they can ask. So we will cover those that are asked in the Terengganu set here. Okay, so I will cut right to the chase. Maksudnya, kita terus jawab soalan, guys. Let's look at the question straight. So this is fermentation, yeast fermentation. Form 4 bab 7. Form 4 bab 7. Yeah, Form 4 bab 7. Okay, now let's move straight to the question. So guys, because I am giving you the idea of how to answer, okay, what I'm trying to say is that the value that student A will be different, obtain will be different from student B, student C, semua angka yang berbeza. So sebab tu, because of that, I will just give you alphabet lah. Let's say this one here, you check how many air bubble. Let's say this is X, this is Y. Okay. Normally, air bubble, because it's very subjective, so they'll give you a range. They'll give you a range. Okay. Let's say they allow you to write something between 10 to 20. They must give a range. Because if not, a funny student put a funny number. Oh, 100. Cannot be 100, guys. Cannot be 100 bubble in 7 minutes. So they apply a range, which they find is very logical. Okay, so from here, you should get 2 marks. I know, around here, you will ask, sir, can I manipulate the numbers a bit? The answer is yes, but don't overdo it. Like I said, don't put 100. Example, don't put 100. Put a very logical number. And if you have the time, just redo lah, guys. If it's quick, just redo lah. Then get the average. I think that's a very safe thing to do. Okay. Next, calculate. So this is what I meant by the value, the first question. Okay. Okay, here they ask you, calculate the rate of yeast reaction for boiling tube A and B. Okay, for boiling tube A and B. So here, based on your table, so for boiling tube A, X number of bubble divided by 7 minutes. So you get another value lah. I just put P lah, let's say P, P per minute. P per minute, okay? Means the number that you obtain here, divide by 7 minutes. So you get like every minute how many bubble secara purata. As for boiling tube B, so guys, what should the alphabet or number be lah? What should the number be? Y, over 7 minutes, I put Q. Q per minute. So let's say these two correct, you should get another 2 marks. Tada, guys, inference. Okay, I think inference is one of the tougher questions to answer for student. I think if I'm a student, inference would be the toughest question to answer. Okay, why? Because they would want, okay, let me teach you how to answer inference. To answer inference, you have to first compare, bandingkan. You compare, then you give a reason. Wow, sir, sound very complicated. Actually, not really. Compare means, like I give you this example. Guys, you know from primary school, they like to make sure there's unity. So they like to put Ali, Motu, and what Akau, Abing, Ahok. I think Ahok. So guys, comparison is what? What can you see here? Ali is taller than Motu. Motu is taller than Ahok. Okay? Or Akau if you like Akau lah, guys. Kau is dog in Hokkien, dog. Kau. No, they won't ask you that in SPM. Okay, so Compare is what? You have to use words like increase, decrease, higher, lower, okay, taller, shorter, but you won't be using taller, shorter. So guys, we say that more for boiling tube B, because there's more glucose. Okay, because there's more glucose, what happens? Now, the compare is more to the RV. If you did learn this in school, inference, even now I tell my students, the comparison is RV. RV is this one. What do you expect of the number of bubbles in B compared to A? Guys, the boiling tube, A more or B more? Answer should be B more. Answer should be B more. Okay, so we say more, or we say a higher number of air bubbles. Okay, a higher number of air bubbles is released. Okay, is released because rate of fermentation, okay, because rate of yeast fermentation is higher. Rate of yeast fermentation is higher. Okay. There are quite a number of answers that you can use because this one is up to your interpretation. Inference means analysis You have to compare, then give a reason. Okay. Sir, I do not want to put a higher number of air bubbles. I want to straight away say more CO2 gas is released. Sir, can or not? Guys, can. No problem. No problem. The good news is nowadays inference is only 1 mark. So biarpun soalan susah, ini hanya guys 1 markah. Okay what? 1 markah. Alright. Okay, next. Variables, guys, as I told you, variables, they will definitely ask. Okay. So variable here, the responding variable would be the number of air bubbles released. Yeah, number of air bubbles released. And by now, you should be very familiar with the method to handle. Guys, the method to handle is different for all MV, RV, and CV. Okay, cara mengendali pembolik ubah berbeza. Cara menjawab berbeza for manipulated, responding, and constant. Now, since we are looking at responding, this one, pembolik ubah bergerak balas, we always start with, normally we start with measure and record. Because here, if you think very logically, measure is wrong. We never measure anything. What did we do? We count the air bubble. So guys, count and record the number of air bubbles released. Okay, count and record the number of air bubbles released in 7 minutes. Like this, yes, 2 marks. Like this, because of these 2, you get another 2 marks. Okay. And as for experiment, guys, experiment, I think, experiment pula. As for hypothesis of the experiment, what's the difference between boiling tube A and B? B got more glucose, right? So we say that the higher Guys, the higher the concentration, of glucose solution, the greater the number of air bubbles released. Okay, you don't have to say 7 minutes because it's a hypothesis. Okay, it's a hypothesis. So like this, you get another 2 marks. Okay, like this, you get another 2 marks. All right. Now, guys, graph, I'll leave it to you. Graph, basically, you should get something like this. I tried it out myself lah. You should get something like this. I'm just sketching one. Okay, just make sure that. Okay, now normally in the SPM, they would have given you the y and x axis. Normally in SPM, they would have given you this, the y and x axis, and they'll label for you the title. Okay, like this one is rate of reaction in yeast. They would have given you rate of reaction in yeast, concentration of glucose solution. So all you have to do is to make sure the scale is correct. Then you just plot the points. Okay, plot the points. Now for bio, we always join the dots and don't extrapolate. Yeah, don't extrapolate to 0. Jangan. Hey sir, half hanging like that. Is it okay? Yeah, no problem. Leave it. All right. So guys, as I told you, they can ask you variables, they can ask you hypothesis. Don't explain your hypothesis, please. Hanya satu ayat, ringkas dan tepat. Okay, so then they ask you to draw graph, and from the graph, they can ask you relationship, they can ask you prediction question. Now we are looking at the prediction question. Okay, let me change color, guys. Let me change color. Okay, for prediction question, a student repeats same experiment as boiling tube B, but does not add paraffin oil. Predict the number of air bubble. Okay, the purpose of adding paraffin oil, okay, is so that kita menghalang oksigen daripada memasuki larutan glukosa tersebut. Now, without the paraffin oil, more glucose can enter. So what do you expect of the number of bubble? Guys, we say that number of bubbles will be more. Or you can say higher lah. The number of bubbles will be more or higher. Okay. Then you get 0. Because prediction, you must always give a range. Guys, you must say more than. Where to get the value? Move back to your initial answer. Ini. Boiling tube B, we get Y number of bubble. So kita mesti cakap lebih daripada Y. Then you get your 1 mark. Okay, so explain. We say without paraffin oil, guys, without paraffin oil, okay, without paraffin oil, atmospheric oxygen can enter the glucose solution. Aerobic respiration occurs and more CO2 gas is produced. By the way, guys, please write in full form. Please write in full complete term. Istilah penuh, carbon dioxide. oxygen. Please write in full. Okay. Guys, another 2 marks. Another 2 marks. And that's it lah. Guys, that's it. If you realize it's actually not that hard, it's just whether you know the concept. So if you look at it, this whole experiment, is it really hard to get at least 12? I think not really, guys, because we have 12 now without even counting your graph. Okay. Okay, so guys, I think this marks the end of this particular pre-recorded lesson. I am thinking of yeah, if I have time, I would try to upload another video to do with maybe percubaan what came out for 2022 lesson for the YouTube students. Okay, just one more small additional help for you guys. Okay, but anyway, for existing students, I'll see you in the usual intensive class. We have still quite a bit more to settle. Okay, we'll be talking about SPM prediction analysis for the past 7-8 years. Of course, percubaan analysis. Then I'll do separately one for the YouTube students. Mostly should be doing a separate one. I haven't decided on that yet. Then finally, what can come out, yeah, and some final tips and so on. Okay, guys, I'll see you when I see you. Yeah, take care, good luck, and just give it your all. Yeah, SPM is just another 1.5 months. Can be very quick, can be very fast. Most important is how you take it. How you take it. Have to have the stamina. Need to have the stamina to endure it. Okay, guys, good luck. Take care. Bye bye.